Okay, I would like to welcome everyone. It's a great honor to be able to present for you my research. You already heard my mentor's, Csaba Pál's introduction to the field of antibiotic resistance. I work in his lab for 5 years now, and today I would like to show you some exact results about my research on the mapping of resistance-free bacterial antibiotic combinations. WHO categorized pathogens by clinical threat, and these ugly guys resulted to receive the tag that critical priority pathogen. So we really fair of them. Namely, these are Acinetobacter baumannii, Pseudomonas aeruginosa, and the Enterobacteriaceae family like E. coli or Klebsiella pneumoniae, antibiotic-resistant strains. So remember the name because you are going to be scientists, doctors, and patients. Hope not, but it is really a high risk that in the future, by 2050, all of these are going to be multi-drug resistant and cause a huge global threat. And though here would have come the good news, but not, in the clinical developmental pipeline, we have just a few antibiotics, these dark blue ones, that has any action on these clinical priority pathogens. So Csaba explained the reasons behind the development of a new antibiotic costs $100 million. And due to the really rapid development of resistance, this cost will not return to the companies. So basically big pharma has no interest in antibiotic development due to this issue. So our aim was to investigate if resistance evolves or not against these new antibiotics under clinical pipeline or clinical development, which has action on critical priority pathogens. See what determines the pace of this evolution and give some help or insight to the doctors or future doctors who you will work with these antibiotics in the clinics. How should we use these new antibiotics to reduce or slow down the resistance? For this aim, we checked the list of the developmental antibiotics, and at this very moment, we selected 7 drugs from 4 different antibiotic families. We plan to add more, and we have them in the lab. We need time to process the experiments with them. So we have these 7 drugs at the moment from 4 different families, which we added for antibiotic resistance evolution experiments, and we also added 1 control antibiotic, which are already well known in the clinics, and they use it excessively to be able to compare the new ones to the old ones and validate our results. We also added 2 strains of 4 bacterial species, all of them from this critical priority ugly guy group. 1 wild type and 1 multi-drug resistant pathogen. I abbreviated them as WT and MDR. You will see these in the next slide. And before going on and see the results, we have to discuss how resistance can evolve. Csaba explained you, if you paid attention, what kind of mechanisms can lead to resistance, like new function enzymes, modified target structures, but how can bacteria reach these new functions? But many of you knew this process in the roundtable discussions and in the laboratory visits, basic mutations in the genomic DNA. This is what we call vertical evolution. Mother bacteria gets a mutation and will inherit this to the children bacteria. Also, gene overexpression is a method, how, for example, 1 antibiotic degrading enzyme can be overexpressed and this way cause resistance. This is also inheritable in vertical way. But big pharma and these drug developers sometimes forget about the horizontal evolution, which is based on conjugation, transformation, or bacteriophage transduction. We in the Csaba and the Kincses Bálint lab, who work in a really strong relationship, can examine all the processes, how resistance can evolve, and we establish the pipeline. And we examine the evolution of resistance against these new antibiotics through all the possibilities I showed you here. Today I am going to talk about the single genomic mutation based resistance evolution against these new antibiotics. That was my part in this very big project we are working on now. But how we measure sensitivity or resistance to antibiotics? Those 10 guys who visited our lab could see how we measure this minimum inhibitory concentration, which is the data for us. And this is the lowest antibiotic concentration that can inhibit bacterial growth. We use these 96 well plates, not these glass tubes that you use in high school, but these tiny plastic holes to examine bacteria, and we create a gradient of the antibiotic with serial dilution that you can see on that side. We inoculate then the bacteria, let them grow for 1 day, and then we can read what is this minimum inhibitory concentration, or MIC. I will use this term also. What is the MIC of the given strain? And if we see increase in this minimum inhibitory concentration, then we can conclude that some resistance occurred, some mutation or some process happened in the bacterial cell. So how I studied the single mutations effect on the evolution of resistance. This is a really simple microbiological assay called frequency of resistance assay. I created huge bacterial populations of 10 to the 10 colony forming units, so 10 to the 10 living cells was in the huge barks. And I inoculated these 10 to the 10 number of cells to antibiotic containing agar plates that contained the 2, 4, 8, and 20 times the minimum inhibitory concentration of the given antibiotic and bacteria pairs. So we measured this minimum inhibitory concentration for all 11 involved antibiotics and 8 involved species, so it was a huge logistics and work. And we performed these experiments with those pairs where this minimum inhibitory concentration was not higher than 4 microgram per milliliter. And then we got resistant colonies after 2 days of incubation. I isolated the colonies from those plates that had the highest antibiotic concentration. I measured the MIC of this mutant population, and then I could have the conclusions about the patterns of the resistance evolution of these 8 strains to these 11 antibiotics, the 7 experimental and the 4 control ones. I used 2 terms in the evaluation of my data. One is the MIC fold change. So how big step could be reached by a single mutation in terms of resistance. So maybe the basic MIC of one combination was 1 microgram per milliliter. After this experiment, I got like 4 microgram per milliliter as MIC for the mutant population. So the MIC fold change was 4 because that was a 4 times increase in MIC. This correlates with the speed of the evolution of resistance. So how fast they can go further to highest antibiotic concentrations. The other term I used is the absolute resistance, so the absolute MIC of the mutant population. So here you can see a heat map of all strains and antibiotics in this matrix. I used the green color for those that were excluded due to the already high MIC. I have to highlight your attention to this line. The multidrug resistant Acinetobacter baumannii, the ugliest guy in our lab, was susceptible just to eravacycline, just 1 antibiotic from our list. So the evolution of resistance is quite high, and so the MIC fold change, and there was just 1 antibiotic which had any effect on this bacteria. But you can see that this yellow color indicated in the given pair big MIC fold change, so rapid evolution, but we have this, you see like blackish color, this dark one. There were no MIC fold change at all in our experimental setup. So those are antibiotic resistance-free combinations, which has a great therapeutic implication, and we will talk about this in the future in some minutes later. Here you can see that the new and the old antibiotics has no difference in the pace of the evolution of resistance. So by MIC fold change, we couldn't detect any significant difference between the old and the control antibiotics. So the developmental directions were not having new resistance-free antibiotics, but something else, like better pharmacokinetical or pharmacodynamical profile, like better side effects, for example. And that was a really interesting result that while working with E. coli, this lab animal of microbiology, we could experience significantly lower MIC fold change. And this targeted our attention to species-specific processes. After evaluating the pace of the evolution of resistance against these new antibiotics and seeing these really interesting patterns, I also wanted to see if my resistant populations are clinically relevant or not. And in previous years, I would use the UCAST clinical breakpoints. These are an empirical data and thresholds. Basically, if our resistance was higher than this threshold, then we could say that it is resistant. If lower, then saying susceptible. But these are experimental antibiotics, so we don't have this data in our case. So we had to use the peak plasma concentration of the antibiotic during the therapy to somehow estimate if our resistant population has clinically relevant resistance or not. And here you can see this key figure of my talk. It seems to be a bit complicated, but it's really simple. I will explain you. In the x-axis, you can see the MIC fold change, what we already discussed. And in the y-axis, you can see the clinical relevance. I put this line, this black line, to indicate the clinical relevance threshold based on our calculations with peak plasma concentrations. So those dots that are above this line are clinically relevant bad news, and below the line, clinically not relevant, so-called susceptible combinations, even after this evolutionary experiment. And by our suggestions, we should use those antibiotics to a given species where the data point is saying that in a bad way, but still understandable, are in this left down corner. So that induced low MIC fold change that didn't reach the clinical threshold. So combining the clinical relevance and MIC fold change data, we could give to a patient such antibiotic that doesn't induce resistance. Thus we could save a bit more time for our antibiotics to be able to use and treat patients. So for example, for Escherichia coli, we could use telafloxacin or moxifloxacin, the fluoroquinolones, showed really good resistance profile against Acinetobacter, for example, doxycycline, or against Klebsiella, sulopenem, a new carbapenem. So we say that using antibiotic resistance experimental data would be able to save us a bit more time to use these antibiotics. So to sum up my presentation, we can see that the evolution of resistance is very rapid against all new antibiotics in at least one or two species. So none of them are resistance-free, sadly. The pace of the evolution of resistance against the new antibiotics is quite the same as the control antibiotics, so what we use in the clinics, and we can see species-specific patterns, which raises the attention for species-specific therapies using antibiotics in the future to slow down the resistance and give a bit more motivation for pharmaceutical companies to develop antibiotics and save our life and be able to live happily in 2050 and don't drive the world to the antibiotic crisis after a pandemic of viruses. So thank you so much for your attention. I also thank for my team and especially Csaba Pál, my mentor, Petra Szili, former Szent-Györgyi student, and Leila Daruka, my greatest colleagues who we work in a really good ambient. Also, I would like to thank all the support I get to be able to conduct research beside university, and I also thank your attention, and I am open for your questions.
And then you trusted the academic program, and I do believe that after these 5 years you can confirm that this program was joining in. However, because the time here is usually we don't have questions in this presentation, however, during the gala dinner, if anybody has any question toward Martin, either in scientific matters or concerning the other program matters, don't hesitate to contact him. And now I would like to ask András Varró to provide the Excellent Student Prize of 2025 going to Márton Czikkely.